Good afternoon, everyone. And we probably all know soil is base for pasture growth, but not every of us know soil probably is the most diversity habitat in the world. When we dig a spade of soil out, you might find some beetles, some earthworms, but there is lots of microbes living in this soil, which we cannot see by eyes. So the soil microbiome is really referred to all the microbes living in the soil, including bacteria, archaea, fungi, and virus, as well as those abiotic and biotic environment factors. So even though they are very, very tiny, but they are quite important. As early as 1966, David Jackson has already described soil microbes as the eyes of the needle through which all the organic matter entering the soil must pass. And more recently, they have been described as the littlest farm hands to indicate their important function. So what can they do in soil? They can decompose organic matter. They can cycling all different sorts of nutrients. They can regulate soil carbon, and their body actually locks the carbon in the soil. Kate referred to it already. And some other benefits is like supporting soil structure. They can produce some hormones to promote plant growth. They can do some pest controls and enhance the nutrient and water take-up, for example, through mycorrhizal hyphae. Kate also mentioned as well. And when the condition right, and some of the soil microbes do release greenhouse gas. So this is a complex system, which Kate already described in a way, and lots of things can influence soil microbes. If we broadly split them, those are three groups. They're in soil environment, plant host, as well as all the biological members living in this community. So you can see lots of factors can influence in this system. So it's a very, very complex system. As well as we know there is a spatial and temporal variability. So the point I want to make is soil microbiome, this discipline, is just at the beginning. And compared to lots of other disciplines, for example, plant breeding, we heard earlier, and this targeted manipulation of soil microbiome for improved pasture resilience, it's still lots of challenges, but we start seeing lots of opportunities as well. For example, using Rhizobia to improve the legume biological nitrogen fixation. We know there is Rhizobia, white clover Rhizobia, present in most of New Zealand pasture sites, but a lot of time they're present in very patchy patches. You see those red dots here, if I can find it's disappeared. So those red dots are the high nitrogen fixation ability, but the blue ones, which is in such a small paddock, is pretty low nitrogen fixation. So if we can inoculate a good Rhizobia into the white clover here, you might see a huge improvement of the clover growth. And some other research our research also showed some soil properties can influence the Rhizobia efficacy in the soil. For example, this graph showed the pH, where the nitrogen fixation efficiency actually increased when the soil pH increased. We don't know the mechanism yet, but might be indication some farm practice can influence this thing. We heard a lot of the climatic stress. Drought is probably the most I heard. So the stress-tolerant Rhizobia, such as the drought-tolerant strain, probably is not only important, but very necessary in the near future. So we have some of the drought-tolerant Rhizobia strain in our collection too. So the other opportunity to utilize this soil microbiome is for plant root disease suppress. Pasture root disease is often underestimated simply because we don't see the root system that often. And when we notice the problem, often it's too late. the pasture looks all right from the above ground, but sometimes when we dig out the roots, and you see those tiny root systems here, as shown in the picture, of course, it impacts on their nutrient uptake as well as water uptake, and eventually it affects the yields. So we have a number of microbial isolates which have antagonistic effect against some plant root pathogens, such as Rhizobia here, and shown in those plates, as well as Pasture. And the other research also showed when the soil changed from disease-conductive effect to disease-suppressive effect, the overall microbial community diversity increases. It's like in those disease-suppressive soil, the plant roots are occupied by those wider microbial community. So protect those roots away from those root pathogens. It's like creating a soil immune response. So we are starting now trying to understand a little bit more how we can promote soil into this disease-suppressive stages by alternation of soil organic matter inputs. The other opportunity I want to cover today is the pest. We heard quite a lot already. Those can be quite damaging the pasture. And Sarah just mentioned in the climate change condition, it's probably going to be worse. But remember, and as for soil-borne invertebrate pests, they have their own pathogens. Those pathogens living in the soils too. For example, Yersinia, and they are quite effective to a group of pasture pests here, like grass grubs, black beetles, piranhas, and etc. Serratia is another bacteria is able to killing the grass grubs. So to maintain those, to control those pasture pests in the soil, so either we need to preserve their pathogens in the soil to a level to keep them under control. The other option is using biopesticide inoculants to target specific pests. So due to the time, I'm only going to cover those three opportunities, but there is a lot more. Hopefully, we will get this paper out soon. But just to mention, soil microbes, they are small, but they do play very important roles in agriculture. It's still very challenging right now for us, but there is lots of potentials for utilizing them for supporting the pasture resilience. We think the future research should focus on those beneficial microbes able to assist plant in response to abiotic as well as biotic stress, and also to optimize those microbiome through management practice. And the other important thing is to provide those scientific validated tools for our farmers. With that, I would like to acknowledge all those current as well as previous programs contributing to this paper. Thank you.